remember way back then, him saying he didn't like cancer treatments where you have to cut people open or hit them with radiation. And he wanted to find ways to get the body's own immune system to fight what it should and not what it shouldn't. In the decades since that bold dream, he co-founded Coley Pharmaceutical Group in 1996, which was bought by Pfizer. He also co-founded the first journal on antisense technology and the Oligonucleotide Therapeutics Society that you see mentioned on the screen. He had been a CSO at Sarepta. After that, he founded Checkmate Pharmaceuticals, which is working on novel oligonucleotides for cancer treatment. He's published over 250 scientific papers and is listed as inventor in over 50 US patents covering oligonucleotide technology. So please give your attention, since you can't give clapping or smiles, to our speaker, Dr. Arthur Krieg. Art?
Thanks very much, Eric. And I won't be talking to you about cancer immunotherapy today. I am going to talk instead about something that I'm not actually working on actively myself, but some of my work has laid the foundation for it. And that is how this area of oligonucleotide therapeutics can be used against the coronavirus. And to start, I just want to go back to the basics of biology, that DNA is the genetic code, DNA is transcribed into RNA, and then that's translated into proteins. And in the DNA, there are 4 bases that make it up, nucleotides. These are the letters of the genetic code, A, C, G, and T. And they have very strict rules of how they form this double helix. C can only bind to G, and A can bind to T. And so knowing those rules and knowing how this works to make RNA from the DNA and then proteins, if you're given any DNA sequence, we know exactly what RNA can be made from it, and we can figure out the proteins that can be made from that too. Now, the human genome is really complex. There's 3 billion nucleotides in our genome. And developing the technologies to read the first human genome took over 10 years, and it cost more than $100 million. That was completed in 2003. But since then, the technological improvements have gotten us to the point where now we can read an entire human genome in less than 2 days for just a few $1,000s. And viruses are much simpler than the human genome. And for example, the coronavirus genome is less than 30,000 bases long. So compared to the human genome, this is much quicker, cheaper, and easier for us to decode. Now, the tools of understanding how to read DNA, we can also write DNA. And this theory was developed, this idea came about in 1978. The idea was to synthesize a piece of RNA or DNA that could bind to RNA and shut it off based on these Watson-Crick rules that we can predict exactly what sequence to make to bind to one single RNA without binding to anything else in the cell. Now, in 1978, when this was first done, to do one experiment, it took 6 months to synthesize enough of this piece of RNA, and this is called an oligonucleotide, to do a single experiment. And so that was not practical. That really didn't go anywhere. And in the 1980s, however, a new chemistry was developed that made it possible to automate DNA and RNA synthesis. And so then it became possible to have, you had these synthesizers, which can fit on a desktop, and you simply type the sequence of the A, C's, G's, and T's that you want to synthesize into this oligonucleotide, and you hit the start button, and then you go off and have a cup of coffee while the machine just generates this oligonucleotide for you. And that did lead to companies starting to try to develop this as drugs. And the first one was approved in 1998. But it really didn't work very well. And the second one, it took 15 years until the next one was developed, because the technology at that point was not very specific. It was not very selective. It really required many further advances to get this great idea to the point where it could actually work routinely. But in the last decade, many discoveries have advanced this. And in just the last 4 years, 8 oligonucleotide drugs drugs have now been approved. So it's taken decades, it's taken literally billions of dollars of investment in basic discovery, academic researchers at universities and scientists working at biotechnology companies in order to develop the technologies to get us to the point where we can now do this on an industrial scale. And I just thought it might be helpful for those of you who have a chemistry background. This is what the natural DNA and RNA in our bodies looks like. This is the chemical structure. And that is not good for making a drug normally because it's very rapidly degraded in our body by nucleases that chew this up. And so one of the first innovations that was developed was just putting a sulfur atom in place of one of the oxygens on the backbone here. And that stabilizes this against degradation. And there are many other modifications, methoxyethyl, fluorine, 2'-O-methyl, of the sugars. Literally thousands of chemist years of work have gone into finding ways to make this work better, finding designs of how to assemble these into molecules that will very selectively just do what you want them to do without these off-target effects. And in the current state today, there are now more than 140 oligonucleotide therapeutics that are in clinical development. And so we're going to be hearing a lot more about these drugs and what they can do. And I just wanted to give an example of one of the technology platforms that is applied here, because I think it's a really good illustration of how science works and how basic science in an area that you think has no practical use can actually turn out to be extraordinarily useful. And that's the discovery of RNA interference, which came in 1998. And this was discovered in worms by Craig Mello and Andy Fire, working in a kind of worm called C. elegans, which, I mean, most people have never heard of that, and you'd never think that this could have any relevance to human health. But it turns out that this mechanism that worms use to defend themselves against viruses, the machinery of that is still in our cells today. Evolution has maintained that. It's used for other purposes now, but we can harness that by making a synthetic RNA that is designed by these Watson-Crick base pairing rules that we understand very well to go into the cell, to be loaded into this ancient machinery that's left over from worms, and that machinery shepherds this synthetic RNA through the cell until it finds the RNA sequence that it's designed specifically to recognize. And when it recognizes that, the machinery cuts that target. And it's catalytic, which means that this process happens again and again and again. And this technology has now gotten to the point in humans where 2 doses a year can shut a gene off without hitting any other genes in our body. And there's a drug called inclisiran for treating high cholesterol that appears that it's going to be safer than existing cholesterol drugs and work better. 2 doses a year of that drug can knock your cholesterol down without apparent side effects on other areas. Now, this target for this drug is in the liver, and we know how to make this technology work in the liver. We'd like to be able to do this in the lungs of people infected with viruses like the coronavirus, because in principle, we could harness this antiviral defense. That's what it originally evolved to do. We can target that against the coronavirus. The challenge is, can we get these synthetic siRNAs, short interfering RNAs, can we get them into the lungs? That's the challenge. There are biotech companies that are working on that right now. This is not going to be solved, I think, in time for this pandemic. But if we develop the technologies to do that against this coronavirus, we'll have them available for future pandemics. Because I don't know if people are aware of this or not, but this coronavirus, there are more than 10,000 other coronaviruses out in the wild, in bats, in all kinds of other species. And most of those are never going to make the jump into humans. But it's statistically inevitable that some of them will. And some of them may be worse than the coronavirus that we have right now. So I think if nothing else, this current pandemic is a wake-up call to us that we need to develop other technologies that can be used against this faster and more specifically than the technologies that we have right now. Let me just say a couple of things about targeting DNA, because there are ways to target DNA with oligonucleotide therapeutics. Gene therapies are one example of that. And gene therapies hold tremendous hope for treating human disease. They're not easy to develop right now, but the technologies are getting better. There are also gene editing technologies that can fix mistakes in DNA or mutations and treat diseases that way. Now, there's a lot of misinformation on social media, and you as fact know that better than anybody, because that's your mission is to, or at least a part of your mission, as I understand it, is to try to counter that. And a lot of the information I've seen on social media is claiming that RNA and DNA vaccines, which is a technology I want to talk about today, alter DNA. They do not. We know how to alter DNA with oligonucleotide therapeutics, and we know how to avoid altering DNA. And the technologies that are being developed for vaccines against the coronavirus do not alter DNA at any rate that is detectable above the natural background mutation rate of DNA, because everyone in their lifetime has hundreds of new mutations that occur. So let me now turn to vaccines against the coronavirus. This is what the coronavirus structure looks like. And the key protein that we worry about for vaccines is this spike protein on the surface. This is the protein that binds to human cells and introduces the coronavirus into our cells to start the infection. And there's a particular domain on that spike protein called the receptor binding domain. That's the part of that protein that has to bind to the receptor in our cell in order to get into our body. And so if we make an antibody that binds to that receptor binding domain, the virus can't infect us. And these antibodies are called neutralizing antibodies. They block the virus receptor binding. That is the goal of a vaccine, to induce neutralizing antibodies. Now, vaccines will also commonly induce the production of antibodies to other parts of the spike protein, because when you have a vaccine, you're basically telling the immune system to respond to what you put into the vaccine. And if you put the whole protein in the vaccine, the immune system may make antibodies against the whole thing. And there have been legitimate concerns raised that some of these non-neutralizing antibodies actually could make vaccines worse. And so this is a major area of research to try to determine whether that could happen with vaccines against SARS-CoV-2. And so far, this is called antibody-dependent enhancement. That's what we don't want, enhancing the viral infection. So far, this seems very unlikely. There was a previous coronavirus that vaccines were developed against, and this was not an issue with that. And early data with SARS-CoV-2 with vaccines in development suggests that that is not going to be an issue, but it's something that the scientists working in this field are continuing to look at. Another question is whether the vaccine should use the entire spike protein or just a portion of the spike protein. And the early data coming out from that is suggesting that it's best to use the entire spike protein. But there's multiple different vaccines in development using either the whole protein or parts of it, so that we're really hedging our bets by, in parallel, developing multiple different approaches. And whatever works the best, that's what we can mass produce. So let me talk a little bit about past technologies used to develop vaccines. And this goes back to the 18th century, the first smallpox vaccine. Initially, the way to make a vaccine was to take a virus that was a dangerous virus and grow it in cells, in tissue culture, until it mutated enough that it became harmless. And this was very unpredictable because there's no design, you're just relying on random mutations, and then you have to test those to see if they're really safe. And it's still a live virus. And so people who have an immune compromised disease or some other condition, they occasionally have serious infections and problems from this. So they're mostly safe, but they take a long time to develop. This is not a useful approach for the coronavirus. Now, years ago, decades ago already, scientists developing vaccines tried to get away from these live attenuated viruses to what are called live attenuated viral vaccines, to subunit vaccines, where you take a protein and then you add something called an adjuvant. An adjuvant is something that stimulates the immune system to make antibodies to that protein. Because if you just inject a purified protein alone without anything else, the immune system ignores it. The immune system has ways of knowing what's dangerous, what does it really have to respond to, and what's not dangerous. And purified proteins are not dangerous. So in the 1930s, when they were trying to develop vaccines against new agents, they just experimented with a whole lot of different things. And they found that aluminum salts would make the immune system produce antibodies to proteins. And that's the adjuvant called alum, which is still used today in many vaccines. Alum is safe, but there's an awful lot of people who are scared of this. And I have to admit, the idea of injecting aluminum salts into your body, it doesn't sound good. We ought to have better technologies than that, and we do. And let me just show you the difference that some of these new technologies can make. This is a standard commercial hepatitis B vaccine that many people get. I got that when I was many years ago. And this vaccine is given on a schedule where there's a dose at the start, and then there's a second dose at week 4, and then there's a third dose at 6 months. And if you look at the seroprotection rate, this is the percentage of people who got the antibody who were protected from the antibody. And with this vaccine, after 2 doses, maybe a third of people are protected. And after the third dose, it goes up to over 80%. Now, that's good, but there's actually new science and new discoveries that the reason why live vaccines work so much better than these alum adjuvants is because the DNA or RNA in live vaccines, depending on whether it's a DNA virus or an RNA virus, our immune system actually detects that as a kind of danger signal. Because even though all forms of life on Earth use the same RNA and DNA basic building blocks, in viruses and DNA, they're modified and arranged in a different way than they are in our own cells. And our body has evolved receptors to detect the viral patterns of DNA and RNA as being different from our own. And these are called toll-like receptors. And there are multiple different toll-like receptors. There are also other receptors, all to detect these patterns in the DNA and RNA of viruses. And when our immune system detects those patterns, it knows this is a virus, and it responds to whatever protein is associated with that. And there's a whole lot of science behind this, just to show you some of the work there. Now, so with a vaccine then, if we come back to this vaccine that uses alum, what if you substitute a CpG DNA? This is a type of danger signal that I have done work on actually for the alum, and you use that instead of alum. Well, this is a vaccine that's now approved that instead of using alum, it uses a CpG DNA that mimics the signal that our immune system uses normally to detect viruses. And if you use that in a vaccine, after 2 doses, you have 90% of people who now are seroprotected. So you've accelerated the development of protection from more than 6 months to 2 months. This is the kind of technology that we need to fight pandemics. This is the kind of technology that can quickly protect people against new viruses. And the safety data also looks good. This type of vaccine has 2 of these have already been approved. We do not see increases in things that people worry about, like autoimmune disease or other things. The side effects of these vaccines are the side effects of your immune system being activated by a virus. You may have fever. You may have redness and soreness at the injection site, maybe some headaches. That's your immune system being activated. And that's through the natural pathways that are used every time we fight off a cold virus or other virus. And so we really don't expect to have any more toxicity from this kind of approach than you have when you get a cold. And you're not going to get the sore throat or runny nose of this because they're not actually any infection. And this CpG has been made available for use in coronavirus vaccines, and multiple different companies are pursuing that approach. So this is a very encouraging advance in technology. And now I want to turn to RNA and DNA vaccines. These have been getting a lot of press recently, and I'll focus mostly on RNA vaccines. The idea here is that we use the genetic code. Once we know the genetic code of the coronavirus, we can manufacture that in a synthetic RNA or DNA, an oligonucleotide, and we wrap it up in lipids and other things that make it look to the immune system like a virus, but again, it's not infectious. And you inject that. And when the immune system sees that with the danger signals built into the RNA that is used in the vaccine, it thinks this is a virus. It makes an immune response against the protein that you put into that, and that can work. And this kind of approach has been developed by multiple companies. I'm showing here some data from a company called Moderna that used this to make a vaccine against cytomegalovirus. And vaccine developers have tried for decades to make good vaccines against cytomegalovirus, and there still is not an approved vaccine. But using an mRNA vaccine at very low doses, 30 micrograms, 90 micrograms, and 180 micrograms are the doses here. And extra strength Tylenol is 500 milligrams, so more than 1,000 times more. This shows you the power of these RNA technologies. After the first dose is given here, the second dose is given here, and by a month after that, you're seeing people at the two higher dose levels having antibodies above what is thought to be the protective antibody level. And this appears to be safe. So when the coronavirus RNA sequence was released, that was in January of this year, that the Chinese authorities shared the sequence of the coronavirus. Within 2 days, Moderna and other companies also worked on this. They were able to design a vaccine based on that sequence. That's all you need. You just need the genetic code of the virus, and now you can design a vaccine that you can have very high confidence is going to stimulate the immune response that you want to stop that virus. And this went into clinical development very quickly. It was about 2 months from the release of the sequence until humans were dosed with this vaccine made from that sequence. In the past, using older technologies, it could take 10 years to get to that point. And so that's why it's possible to talk about developing a new vaccine at warp speed. Because of these technologies, we can greatly accelerate that over what was developed, over what was done in the past. And there is early data out now from the phase 1 clinical trial using this vaccine against the coronavirus after 2 doses. All of the participants so far at the first 2 dose cohorts seroconverted. That means they made antibodies at the level of people who have recovered from infection, convalescent sera. And there were neutralizing antibodies in all 8 of the subjects. So this is exactly what we're hoping for. It's a very small number of people. Clinical trials have to be done in usually over 10,000 people to detect side effects that occur only once in 10,000 people to make sure that it's safe. But we're off to a good start with this. And that's just one of the mRNA vaccines. All of these other companies are in parallel with Moderna, developing other approaches, some of them testing multiple approaches in parallel. So there's a lot of work going on here, and I think there's reason to be optimistic that some of those approaches are going to succeed. Another type of vaccine that you may have heard about in the press is called a viral vector. And the Oxford University vaccine uses this approach. The theory here is to take the DNA code for the coronavirus spike protein, and you essentially splice that into a harmless virus. Adenoviruses are a common type of cold virus that are used for this purpose. They're also used for gene therapy as a way to introduce new DNA into cells. And this approach also can work for a vaccine. After the first injection of these vaccines, though, you make antibodies to the adenovirus itself that neutralize it against being able to give the vaccine again. So remember, I showed with the Moderna vaccine and with other vaccines, you typically give an initial dose, and that's the priming dose to start the immune response. And then you give a booster dose. And it's really only after the booster dose that you get the strongest immune response. These vectors work very well, but you only get to give one shot. And so you really can't expect to get as strong a response after just one shot as you can get after several shots. But these are moving forward in clinical trials. And the vector used here has been tested before in humans against a different coronavirus. And there was some activity. It wasn't great. It was less than half of people at the highest dose who made neutralizing antibodies. And in the monkey study that was done using the Oxford vaccine against the new coronavirus, there were 6 monkeys in that. There was partial protection of the monkeys. They still got infected, but it was milder than what you would have expected. So it's better than nothing. And this is also moving forward in development very rapidly. And I think it will work. I'm just not sure that it's going to work as well as a vaccine that you can give booster doses. So, in summary, I think that RNA vaccines can be developed against many viruses very quickly because of these new technologies that have been developed. And if it's a type of virus that we've had experience with before, and we have had experience with coronaviruses before, the spike protein is reasonably well known, and we've had success with blocking spike proteins of other coronaviruses using this approach. So I do expect to see several safe and effective vaccines against SARS-CoV-2. They could be the protein subunit approach plus a synthetic CpG DNA. They could be the mRNA vaccines, the viral vectors. All of those different approaches are being tested. Probably not all of them are going to work equally well, but I think we will, toward the end of this year, early next year, we'll start to have doses available because the manufacturing is being ramped up now at risk to prepare for this. And after this, as I mentioned before, there's a lot of other coronaviruses in the past. We need to learn from the mistakes that have been made in responding to this current pandemic so that we do better in the future and that we can be prepared for those. And finally, I do want to mention there are some exceptions where efforts to develop vaccines have not worked. And so we need to also learn from the experience where this hasn't worked. HIV is a retrovirus. That's a different kind of virus that's very difficult. Hepatitis C, hepatitis B, dengue virus. There are reasons for each of those why they're so challenging to develop vaccines against. And I don't think they reflect negatively on our chances for a vaccine against the coronavirus. So I'll stop there, and I'm happy to take questions.
Okay, I'm Becky Strickland, I'm the moderator, and our first question is from Sanjay. In one slide, you showed how in a worm, its cellular mechanism snips and modifies the RNA of an infecting virus that appeared as CRISPR-like. How do non-CRISPR-based oligonucleotide therapies differ from CRISPR techniques? Yeah. Is one technology, there's more, is one technology better? For example, more robust, faster time to market for addressing the current coronavirus?
Yeah, great question. So RNA interference, which is the one from the worm, that works at the level of RNA to attack the RNA of a virus. CRISPR technology originally was developed to work at the DNA level. So you could, in theory, use a CRISPR approach against DNA of a virus, but they're complementary. The trick with the CRISPR approach, though, is we don't want to change our own genome, or it's risky. It's really risky to edit DNA, because if we edit our own DNA and we introduce a mistake, in theory, we could cause more problems. And so I think it's safer for fighting viruses to stay away from genome altering technologies until we understand them much, much better than we do right now, and instead attack them at the RNA level.
Okay, next question is from Doriva. It's great to hear there's so much advancement in the vaccines. I'm sorry, somebody put another one in there, so things bounced around. Can the vaccines you are citing here be created quickly enough, at scale, and at a reasonable cost to cover the needs of the world, not just the developed countries, when they are ready?
Yeah, that's one of the key questions, because originally what Moderna was talking about originally, for example, was that maybe by the end of this year, they could have 10 million doses. And that with support from the government to scale up manufacturing, they might be able to make 10 million doses per month during 2021. And when you do the math on that, you can see, well, that's not even going to be enough for the US. What they're saying more recently is, well, first of all, Pfizer, Sanofi, all the other big pharmas are jumping into this, and maybe I shouldn't say all of them, many of the other big pharmas are jumping into scaling up manufacturing to be able to produce hundreds of millions. And I saw the other day, I think it was Sanofi said their goal is to be ready with 2 billion doses of coronavirus vaccine in the next year. So as I've been doing the math, adding up what all the different companies are saying they're going to be able to produce, we could get to the point of having 7 billion doses sometime in the next year or so, or toward the end of 2021. And I think that's obviously one of the key responsibilities for having these technologies, is finding ways to get them to the people who need them. And once we have this manufacturing capacity established, once we've made the plants that can manufacture hundreds of millions and billions of doses of these vaccines, we'll have them ready for future pandemics too, so they can be rapidly applied to that.
Okay. Next question is from Nick. Nick, I'm not sure I'm pronouncing your last name right. Kaiser. mRNA vaccines require that the cells that take them up translate the mRNA into viral proteins, which are then recognized by the immune system. Is the timing and strength of the response to these vaccines different from vaccines based on viral proteins?
Yes, it is. And that's one of the tricks to them that I kind of glossed over. Because what happens with the mRNA, because it's mimicking a virus and because it contains these viral patterns that are danger signals that are detected by our immune system like a virus, the virus reacts to that by making interferons. And interferons, if you have too high a level of interferons, they shut down the translation of the viral protein. When you're giving a set measured amount of a viral protein in a subunit vaccine, you know exactly how much protein you're giving, and the immune system makes a response to that. If you don't design the mRNA vaccine correctly, if you design it and it induces too much interferons, they can shut down the production of the viral protein before the immune system makes a strong response. And so in the development of these vaccines and in the testing of them of humans that's been done so far, one of the key goals for the researchers was to tweak this and to design the RNA to have enough of the danger signal so that the immune system will respond to the viral protein, but not so much that you just shut the system down. It's a key question that I have, and I think it's one of the things that has to be tested across thousands of people, because no vaccine is going to work in 100% of people. Even the best vaccines only work in maybe 95% of people. And when you get to the elderly, people infected with HIV, people with diabetes, response rates are often 50%. We need to find out with these new vaccines, how well do they work in all of those different populations, and we don't know that yet.
Okay, the next one is from Scott Snell. of National Capital Area Skeptics. Hi, Scott. Thank you for your excellent presentation. Do you have any comments or concerns about the expedited process being used to develop and test the current vaccine, COVID vaccine?
Yeah, I don't see that we have an alternative to this. I think in this condition, we should move forward as quickly as we can without sacrificing safety. And I think we can do that with these technologies. I mean, the one thing that we won't be able to do is to have years of follow-up with seeing what happens in 10,000 people who get the vaccine over the next 3 years. And one of the concerns, one of the questions that people have about vaccines is always things like, what's going to happen long term? What's this going to do to my immune system? Am I going to get an autoimmune disease? I trained as a rheumatologist, and when I started working in this field 30 years ago, I actually thought that activating the immune system with DNA or RNA was going to cause an autoimmune disease. And it turns out that it doesn't, at least at any level that we can. But normal infections. So during our normal lifetime, you get thousands of infections. And every time you get one of those infections, they activate your immune system through these natural pathways, these toll-like receptors, and your immune system fights off the infection. And most of the time, everything goes really well. But 7% of the population has some type of autoimmune disease. And in a lot of those people, there's evidence that that autoimmune disease could have been triggered by an infection that they had. And what happens when you're fighting off an infection is your immune system is trying to just fight off the infection without attacking the body's own tissues. But occasionally it goes wrong. And so activating these pathways in a vaccine, we can expect that occasionally something could go wrong, just as it does when you're fighting off a normal cold. The difference is when you're fighting off a cold or other virus, the virus can actually get out of control because it's growing. And so it can overwhelm the immune system. When you're giving a vaccine, it's not something that's growing. You've designed this and you've measured a dose that is not going to overactivate the immune system in the way that we can detect. So the way I think about it, the way I explain the risk to people is that the risk of these vaccines is the same as the risk of having a mild cold, because these vaccines are activating the same pathways that your body uses to fight off a cold virus, a harmless cold virus that you fight off many, many times in your life. And 99.99% of the time, nothing's going to go wrong. In a study of 10,000 people, these types of vaccines against other agents have already been tested in more than 10,000 people, and we see no increase in autoimmune diseases in the follow-up time from those. So any added risk must be way below your lifetime risk of having an autoimmune disease, which is over 7%, because over 7% of the population has that.
Interesting. The next question is from Linda Blythe. Will the virus mutate, causing us to need a new vaccine every year like we do with the flu?
That's a concern, and it could, and I think we need to be prepared for that. The good thing about these types of technologies is that we can change them very rapidly if the virus does mutate. And the good news about coronaviruses is that unlike cold viruses, coronaviruses have a proofreading mechanism built into the virus that reduces its mutation rate. And for us, that's good because these viruses do not seem to mutate as fast as cold viruses do, but they do still mutate. And also, as I mentioned, there's a heck of a lot of other coronaviruses out there. And so we do have to be prepared for other viruses to come along.
Okay, next question is from somebody named Eric Krieg. Hi, Eric. How long do you think it might take for oligonucleotherapy to be common in the fight against different types of cancer?
Well, that's something that we're working on, and I think that it's going to be Well, I'm optimistic. I always think things are going to take less time than they end up taking. I think in the next few years, we'll start to have more treatments approved for treating cancer that can harness some of these same immune pathways that evolved to fight viruses. And we can apply those to essentially trick the immune system into thinking that a cancer is actually a virus. Because the same defenses that can fight off viruses, and we can apply those defenses to fight off cancer too. I think we have had some success with that already that we've presented at scientific meetings, and we're getting better at learning how to do this. And so I think the future of cancer immunotherapy is very exciting and very bright.
Next question is from David Cattell. Do viruses ever mutate so that they become harmless to humans?
Yeah, we tend not to know that, because if the virus becomes harmless to humans, we don't detect it because we don't know the people got infected. But there is very good evidence that this can happen. And that was in Australia. They had a rabbit plague. And so they imported a kind of virus, I think it was paramyxovirus, I think it was, that is 99% lethal to rabbits. And they imported that into Australia. And immediately it wiped out 99% of the rabbits. But the virus mutates at a certain rate. And the only rabbits that survived were the ones where there was a milder form of the virus. And so pretty quickly, the form of the virus that was circulating was completely harmless to bunny rabbits, and they all bounced back in as big numbers as before. And now they were immune to the virus. So that kind of thing can happen.
And maybe work against us.
It could.
Yeah. Okay. Next question is from Joe Walsh. It is hard to make predictions, but what do you see as the state of infectious disease and vaccines in 100 years?
Wow. Gosh. That's hard to imagine. When I look at the progress that we've made up to this point, I would like to believe that we're not going to have any infectious diseases anymore. Have the technologies to our immune systems so that we're just completely invulnerable to infection or Alzheimer's or other conditions. Salamanders can regrow limbs. Why can't we do that if we really figure out how to control the genes at that level? It's not going to be easy, but in theory, it ought to be possible. So I'm an optimist.
Okay. We'll have to wait and see. Next question is from Robert. Is there any reason to think the immune response to an mRNA vaccine would be any different in neonates and infants compared to older children or adults? Asking
Yes.
a pediatrician.
Yes, absolutely. And in fact, I was just talking to a pediatric immunologist last week about this, because as you may be aware, the coronavirus doesn't seem to be as severe in most children compared to adults. And the immune systems of children are different than in adults. They're biased in a different way. So that's one of the things that we have to test. And I just don't know now whether the whether the vaccines will work just as well or better or less well. But some of these toll-like receptors function differently in kids. There's one called toll-like receptor 8 that seems to dominate more in infants than it does in adults. And we just have to do the testing to see, will the vaccines work as well there?
Okay. Next question from Scott. Why don't we have a vaccine for SARS or MERS, MERS?
Yeah, they were developed, they were tested in humans, and they did have some activity, but there was no funding to continue the development. So the good thing is that the testing was done, and based on the experience from those, relationships were formed between the National Institutes of Health and various companies here, and groups like BARDA, which is one of the government agencies involved in biodefense. This was already on their radar screen, and they had already been anticipating future pandemics and preparing to develop the technologies for this. And so once the coronavirus came along, some of the executives and scientists at these companies that had studied the MERS and original SARS coronavirus called up people at NIH and other agencies and said, hey, you know, what do you think? Should we start working on this? And the answer was yes, and funding started to come in from multiple different groups. And because we already had that experience, it really made it so much faster and easier to do this against the new coronavirus.
Good. Okay, another question. Would you comment on the talk of antibody therapy?
Yeah, I think antibody therapy is great because you can make recombinant antibodies that are going to protect you against this. And there was just a study that came out the other day, actually, showing that people who've recovered from coronavirus, you can actually identify neutralizing antibodies in their blood, and you can you can mass produce those antibodies. Now, there's two types of antibody therapy that you should be aware of. One is called convalescent serum. And that's where you take someone who's recovered from infection, and they presumably have the antibodies in their bloodstream, and you take their plasma, which contains all of the antibodies in your bloodstream, and you then give that plasma to somebody else to protect them against infection. And basically, you get enough antibody from one person to treat one person. Whereas if you mass produce a single defined neutralizing antibody, you can manufacture enough antibody to treat tens of thousands, hundreds of thousands of people. And so the focus, the near-term focus until we can get the mass-produced antibodies is just convalescent serum, which works at a small scale. But the large-scale mass-produced antibodies are going to be, I think, very helpful. And one of the ways that they could be helpful is making it possible to safely do human challenge studies. Because how do you show that a vaccine actually works to protect humans against infection? There's two ways you can do it. One is you can just mass immunize tens of thousands or thousands of people in the community and hope that the infection spreads enough that you can see between the people who got placebo and the people who got the real vaccine that there's a difference. Or the other way you can do it is you can bring people into a clinical research unit, normal healthy volunteers and people have already volunteered for this, and you can give them the vaccine and then challenge them with the coronavirus. And you have to have a control group there too. And the only way to protect the control group if they get infected and have a severe infection would be with an antibody therapy. There's really no other drug right now that looks as promising as antibody therapies.
Interesting, interesting. Eric, are you on camera? Maybe not, so I can wrap this up. Excellent presentation.
Well, thanks, Becky.
Presentation.
My pleasure.
Very, very clear and concise. I was nervous myself about whether I would be able to follow. And I did, and incredibly clear, concise questions and clear, concise answers. So thank you, everybody. And I see Eric, so Eric.
Thanks so much, Art. You've done good.
Thanks.
Really.
I'll tell our doctor dad about the good job you did when I call him in another 10 minutes.
Thank you very much. And thank you all for your questions and your interest, and keep up the good work that you do. In fact, I think it's important to have organizations like this.
Thank you. Thank you very much.
Thank you. And everyone join us on June 20th for the same time when we have Paul Offit talk about excesses of medicine. So thanks everyone for joining us for this. Best wishes all. Bye-bye.
Bye, everybody.